everybody, this is a quick video to show you how to do the Respiration Lab, so you can watch this ahead of the lab and make sure you understand what you're doing, or it's a great video to watch before the AP exam just to remind yourself about all the labs that we've done. So this lab is called Respiration. We are using pea seeds, and these pea seeds, you'll see they're, I already lost one. They're already nice and big, they have filled up with water, and they've started to germinate. So germinate means the embryo has started to grow out of them. These are going to become seedlings. So I took some of these seeds, and the lab told me to use 25, but the respirometers we have wouldn't fit 25. So I actually just used 10. And the first thing I did was I put them into a graduated cylinder that already had 20 milliliters of water in it. When I added it, the water level of those 10 peas went up to 27 milliliters. That tells me 7 milliliters is the volume of my peas. And then what I did, because I want to create respirometers that each have an equal volume inside them. For the second respirometer, you use dry peas. So here are the dry peas. They're really crinkly and much smaller than the others. I took 10 of them because I want to have 10 living. They're both living, but these are very active, metabolically active seeds. These are very metabolically inactive. They're in a state of dormancy. Until they germinate, nothing is really going on in these seeds. So I want to use 10, the same number of them, but I want them to occupy the same volume in the respirometer. So to make up the remainder of that volume, I use beads, these blue beads. So what I did is I took the graduated cylinder, again I filled it to 20, I put in the 10 dry seeds, and then I added enough beads until it went up to 27. I poured those out on the table. What's in here, I just did my third respirometer, will have just beads in it. So I'm going to pour out the water real quick. These are the beads that I will need to use that will occupy 7 milliliters. So now I'm using 7 milliliters of stuff. For number 1, they're active seeds. For number 2, they are dry pea seeds. And for number 3, they're just beads. So you might be asking yourself already, why am I going to even bother having one that's just dry beads? The reason why, that is my control. I want to see if any changes occur in my respirometer with the beads. That must be just due to the temperature and pressure changes that are occurring, and I want to subtract that value from my other 2 tubes. So I'm going to use that control to give me a corrected difference. Alright, when you set these up, you want to first use a piece of absorbent cotton. So a cotton ball is absorbent cotton. Take and break the cotton, so you just have a little bit. Use a pipette, put it at the bottom, and then take a couple drops of potassium hydroxide. The reason why we use potassium hydroxide, and you don't want to get it on your skin, it's actually caustic, it'll burn your skin a little bit. You do not want any of the KOH to touch the sides of the respirometer, just the bottom cotton. So that's why we use a pipette. So just a little bit of KOH. KOH will absorb any of the carbon dioxide being produced by respiration within these seeds. So we're really able to measure how much respiration is occurring based on how much oxygen is it taking in, and we're completely not looking at the carbon dioxide produced because it will crystallize with the KOH in the bottom of the container. Now, because I just told you that's sort of a harmful substance, we put a little bit of non-absorbent cotton, because if I put another piece of absorbent, the KOH is going to go all the way through. I put this in. So I take another pipette or a scoopula, something, and I push that down to the bottom. So now no KOH will be touching my seeds. I'm going to set up the first respirometer by putting in these 10 germinated pea seeds. And then this is what I'm going to add to it. A graduated pipette that is sealed with a stopper. So I carefully turn this in. I also make sure that I'm not hurting any of the seeds with the end of this. I will do the same thing for each of these, and I will set them up. When they're all ready, the next step is you take a large container and put room temperature water in it. Then you're going to set up a piece of tape on one end, take your respirometer, set it in the water with the open end on the tape. Let it sit for 5 minutes. We call this an equilibrium period. It allows the respirometer to get to the same temperature as the water is. After 5 minutes, I will have all 3 respirometers in here, and I will let them go down into the water. When the respirometer goes down into the water, a little bit of water will go in the tip, and the remainder of the respirometer will all be air. I will get an initial reading that tells me where the water level is at, and over time, I should see, especially these germinating pea seeds, taking in oxygen. If they are breaking down sugars because they're trying to grow and become a seedling, they are going to need oxygen for that growth. They're going to need a great amount of ATP. So as I watch the water move in, that's indicating to me how much oxygen it is consuming. The one that has the dry peas and the beads, I shouldn't see really any air move in at all. And same thing with the beads. I would expect 0 movement. If it moves in a little bit, I want to subtract that little bit from all of my other number 2 and number 1. That will give me a corrected difference. If they tend to float and not sink very well, just take a washer, put it over the end, and that will keep the respirometer down in the water. I advise you to do that at the very beginning because that way you don't have any airs on the way. If you have any other questions, let me know.